All right. Good afternoon, everyone. So welcome back to NACFC. So I think it's time to let's go ahead and get started. First, thank you all for coming to this. Let me see. Okay, here we go. Okay, thank you all for coming to this cell biology of airway epithelium workshop. I believe it will be a very exciting workshop section, and because of the quality of the abstract. My name is Yunjie Huang, assistant professor at Indiana University. So along with me is Dr. Tom Kelly, professor from Case Western. We will moderate this section together, we don't have anything to disclose. So airway epithelium is the cell layer lining our entire respiratory system. However, it is harmfully affected by cystic fibrosis, leading to airway with acidic environment, dehydrated mucus, chronic infection, and hyperinflammation, and so on. Understanding the cell biology of the airway epithelium is critical to restore the healthy airway epithelium layer in people with CF. As we know, airway epithelium cells are heterogeneous. They are highly differentiated and polarized. They consist of ciliate, not good. So they consist of ciliate cells and goblet cells, basal cells, and you're probably very familiar this famous ionocyte and others. So, so these cells are lining these airway. Loss of CFTR function in CF alter the biology of the airway epithelium cells. Today, this section will present you the study that detail aspect of CF airway epithelial cell biology using different cell models, including nasal cells, and large airway cells and small airway cells. Specifically, the following topic will be discussed: nasal cell. Nasal cells response to the long-term CFTR modulator therapy, and mucociliary transport in hypoxia condition, basal mucus concentration, and epithelium polarization, and novel TGFβ signaling mechanism. So lastly, I would like to remind and encourage our audience to type in your questions through the NACFC app. We will try our best to get your questions answered, as many as possible. So without further ado, I would like to welcome our first speaker, Dr. Esther Velada. She's an assistant professor from University of Colorado. The title of her talk is Sinonasal Epithelium Functional and Transcriptional Response to the Long-Term Highly Effective CFTR Corrector Therapy. Close it.
All right, hi everybody. Again, my name is Esther Blatter from the University of Colorado, and I would like to say thank you for this opportunity to talk about our work, trying to understand the sinonasal responses to long-term CFTR modulator therapy. And before I start, I would like to mention two related presentations at this meeting. The first one is a poster by my colleague, Dr. Dan Beswick, which describes the clinical and patient-reported outcomes from that are associated with the study that I'm going to be talking about. That is poster number 571. and another presentation that I will have tomorrow afternoon where I will present data from a separate single-cell sequencing study that's kind of a sister data set to the one that I describe here. All right, so with that, I have no disclosures, but I have a lot of acknowledgments. So first, I'd like to acknowledge a team of collaborators led by Dr. Jennifer Taylor-Kauser and Dan Beswick. And I would like to specifically highlight my bioinformatics collaborator, Dr. Austin Gillen, and then also members of my lab who worked on this study and then our funding sources. So I will start with the problem that we're trying to address. And I have a long-standing interest in the extensive structural and functional defects that characterize the cystic fibrosis airway epithelium. And these are illustrated by this really nice scanning electron microscopy images that I took back in the day when I was trying to familiarize myself with the disease. We're looking at the apical surface of the turbinates. The airway epithelium looks like a beautiful shag carpet with lots of cilia. And the CF airway epithelium from a single donor, as you can see, is highly variable in appearance, ranging from nearly normal to grossly remodeled, highly abnormal, as you can see by this extensive mucociliary metaplasia. So this is what is remodeling, which is defined as detrimental structural and functional changes. We know that this underlies disease symptoms and drives disease progression. It's focal, highly episodic, and variable in extent. And there may be some features that are irresponsible. And we've been using the sinonasal epithelium as a model, and later on I'll kind of circle back to raise the relevance and the relationship of this to lower airway disease. All right, so with this in mind, I raise three critical questions regarding remodeling in the context of initiating modulators in people with established CF lung disease. So first, we still want to understand the mechanisms that drive remodeling within the CF airway epithelium. Second, we want to know what is the extent of improvement that we can achieve over time. And importantly, how does benchmarks to healthy individuals that are matched in age. And with this, one of the potential role modulators may play in the future is to repair or potentially preserve structure and function until eventual gene therapy. And then finally, very importantly, in the forefront of my mind, are how useful are transcriptomic data, especially those from the sinuses, to characterize and monitor CF disease in the lungs in response to modulators or really any other therapeutic intervention. So while remodeling is complex, mechanisms are not very clear. This is sort of how I think about it in this cartoon, and also how correctors could play a game here. So in CF, we start in early life with a dysfunctional CFTR ion channel, which leads to the buildup of difficult to clear mucus. This starts this vicious cycle of infection, inflammation, epithelial injury, and then through aberrant repair, eventually you get remodeling. And it remains unknown to what extent CFTR modulator therapy, which in this case I'm referring to Alexatez-Iva, is able to rectify these changes, especially when initiated in an adult cohort with already established disease. All right. So as to the study that we designed to understand the clinical, patient-reported, as well as transcriptomic outcomes after 6 and 24 months of ETI therapy in adult cohort. In this study, we had a special emphasis on sinonasal disease. I'm going to tell you a little bit about this study first, but before that, I have to again acknowledge my very generous and incredible clinical collaborators who initiated this study, Jen Taylor-Kauser and Dan Beswick. And the goal here primarily was to test the hypothesis that ETI will lead to improvements in CF-related chronic rhinosinusitis, improvement in patient-related outcomes. And again, I would like to refer you to Dan's beautiful set of publications on the baseline to 6-month analysis in his poster. And then finally, we hypothesized that these changes will be accompanied by improvements, so these clinical improvements will be accompanied by changes in sinonasal gene expression and cellular function. And this is what I'm going to be talking about today. This was the focus of my lab at the University of Colorado. So here is sort of the outline of the study. We collected 22 complete time courses, but several other individuals who were enrolled provided either baseline or other samples. So we sampled individuals at baseline when they essentially picked up their prescription, then again at 6 months, then again at 2 years. The right nasal brush, which we collected, was used for bulk whole transcriptome sequencing and microbiome analysis. This is what I'm going to be talking about today. And the left brush was used to isolate and expand airway epithelial basal cells. And if you're interested in the work from that, please come by tomorrow. I'm going to be talking about that along with the single cell data. So I would like to briefly set the stage with an overview of the clinical outcomes of this study, but for the full download, please go and check Dan's poster. So first, just looking at the demographics of the 22 individuals in the study who participated in the full cohort, we can see that we have an adult population that has established but sort of variable extent of pulmonary as well as sinus disease, and the latter of this is expressed as this sinus CT opacification. So this is a way to evaluate sinus disease by CT scan, and sort of a higher extent of opacification is consistent with a higher extent of disease. So looking at the data, substantial improvement was observed in FEV1 from baseline to 6 months. Here you can see the averages and then the individuals broken out. And this was stable through the 2-year time point. And here I'm actually showing you data from all the subjects we collected data from. And then for the CF-related sinusitis, the same outcome was observed. So let's start with looking at the bulk transcriptomic analysis. These are the number of subjects that we were collected. We carried out RNA-seq on basically everybody that we were able to capture. So what I'm going to be showing you is the data from the all-comers analysis, but if we're just restricted to those 22 individuals, we essentially get the same outcomes. So I'd like to first sort of take you through a couple of technical aspects here. What you can see here is the samples were sequenced in multiple batches, not my preference, but such is life. So first we sequenced the baseline and the 6-month folks, then the 2 years and the extras, and then we went ahead and we resequenced the number of individuals at the same time as a foundation for our batch correction. So you can see a PCA plot here where the sequenced and the resequenced samples show a really beautiful correlation in their gene expression. We also had very, very high RIN scores. Our average RIN score was just a little bit over 9. So we know that we went in here with an exceptionally high quality prep. Regarding the analytical pipeline, we used what happened here? There we go. So we used a negative binomial mixed model pipeline. This was something that's been shown to be very robust in the type of longitudinal sampling and for the sample sizes that we're doing. And then our analysis yielded about 1,000 or 2,000 DEGs, or differentially expressed genes, with an FDR cutoff of 0.05 at each one of the time points that are listed here. And then before we get into the results, I want to sort of put out there or acknowledge that the pandemic has been a huge confounder and complicator to everything that we have all been trying to do within the last couple of years. We have one individual who was sort of computationally suspected to be COVID-19 positive in our sample, although they tested negative. We had to have massive changes in our primary cell handling procedures. And as a cell biologist giving a presentation without any cell biology in a cell biology session, it's important to remind myself and to all of you that transcriptomics is a hypothesis tool, generating tool, and it doesn't replace the rigorous experimentation that should be following that. So with that, let's look at the pathway analysis that describes the major gene expression changes, first at baseline to 6 months. So very, very satisfyingly, if you're looking at the downregulated versus upregulated pathways, our signature is dominated by a massive reduction in neutrophilic inflammation, while the upregulated pathways indicate metabolic changes, organelle biogenesis, including ciliogenesis. So we carried out ingenuity pathway analysis, which shows this network of genes that describe, genes and processes that describe the major changes, with blue showing predicted inhibition and an orange showing predicted activation. And I apologize for the small font and the color scheme changes, but what I did here is to highlight a little bit some of these nodes to indicate to you that what we're seeing here is a reduction in very much the type of inflammation in the nasal airways that has been very, very well studied in the CF lower airways. So now what I'm sort of highlighting here obviously rests on the work of many, many, many groups over many, many years. And I indicated a review here that sort of nicely summarizes some of this work. So the major changes and the major players here include some very, very well-known mediators like IL-1, TNF, interferon gamma, some potentially new players like DOC8, which according to my brief gene cart search has to do with NK cell dynamics. So if anybody's interested in studying DOC8, there may be some good biology there. You can see processes like net formation and signaling, and then finally the increase of pro-resolving IL-10 signaling pathway. And then to help you a little bit with the interpretation, we recover about 15% immune cells, and then the rest of them are epithelial cells with these nasal brushes. So primarily these signatures are derived from a combination of these cell types. Okay? But there's also other strong epithelial signatures as well, including a decrease in the epithelial cortification program. This is a signature of a squamous cell population that we have become very interested in as sort of a dominant, potentially interesting player within a remodeled airway epithelium. And I'm going to be showing some trajectory analysis from our single cell data to try to understand how these cells become expanded within the CF airway epithelium. Okay? We also see the cell type that I've been studying throughout my entire scientific career, the ciliated cell, expanding as CFTR is being corrected, and this is sort of indicated by this big uptick of genes related to cilium biogenesis, ciliary trafficking, and ciliary signaling. Okay, so now onward, we look at the 6 to 2 year changes, and to our surprise at the cohort level, we observed an increase, albeit at a lower level, of the same inflammatory pathways that were so drastically downregulated at 6 months, suggesting, and I'm going to be calling this a transcriptional resurgence of this, again, the same flavor of neutrophilic inflammation. Okay? So here's sort of another way to visualize this. What you're looking at here on my left is a way to show the clustering of the gene expression profiles of the up and down regulated gene lists within the different time points. And what you can see is that the baseline to 6 month down-regulated genes and the 6 month to 2 year up-regulated genes strongly clustered together through these inflammation-related pathways. So this diagram around the circle shows sort of individual genes, and anytime that there's sort of the same gene present in the same list, you can see this sort of connection, and you can see that indeed these are very much the same genes. It's not that folks are, everybody got COVID in our data set in year 2. So this is the resurgence of very much a similar type of inflammation. So obviously, it's important to note that this is not occurring in all of the subjects. When we break down the expression to the subject level, and here I'm just highlighting to you normalized to baseline, the expression to 6 months or 2 years of the ILA transcript, and you can see an increased expression in 2 years in only about half the subjects, and those I marked in asterisk. So again, the take-home message here is that the cohort level conclusion from the patient level data is that there is stable outcomes for this 6 month to 2 year period. However, transcriptomically, we're seeing, at least in some of our subjects, a potential for resurgence. And then obviously the most important question to address at this point is there correlation between the clinical data that we're seeing? So again, we're going to go back to this sinus opacification measure. So this is the primary endpoint in the study. It was assessed on CT scan via a machine learning algorithm. So what you can see here is that this is sort of the opacification that we want gone. These are some beautiful CTs showing that indeed that it does. But if you look at the 2-year response, and I have been sort of looking at people's data at this meeting so far, what we can see here is that these sort of further time points, even if we sort of regress to this sort of stable, no progression cohort level message, it is really composed of individuals who maybe hold the course. Some people get better, some people get a little bit worse. And this is where potentially the interpretation becomes a little bit challenging as to what constitutes a clinically significant change. Now, when we carried out a mixed effects model association with the person SO and the entirety of our baseline 6-month, 2-year transcriptomic signature, what we found is that these same genes that are related very strongly to CF inflammation or neutrophilic inflammation are the ones that had very strong statistically significant correlation to the outcomes as assessed by the sinonasal opacification score. And what we've been doing is that we've been working towards establishing what we're calling a transcriptomic signature of the Trikafta response within the airway epithelium. What we're hoping is that this is going to be an alternative, highly sensitive outcome measure for airway disease. And we're sort of hypothesizing that this may predict or precede clinically detectable changes. So with this, I'll kind of provide a little bit of a wrap-up. So we conclude that we show transcriptomic changes that are consistent with improvements in chronic inflammatory remodeling. We detect a highly heterogeneous response, as sort of expected, and the possibility of this transcriptomic regression in many subjects. And I mentioned that our goal is to derive this robust transcriptomic signature that could be used to continue to study this response in other cohorts. So with this, we're very interested in continued longitudinal sampling. We're very interested in seeing that maybe the correlations will be holding, potentially clinical changes will also be reflected in what we're seeing transcriptomically. So we're in the process of validating this signature with other validation cohorts, both in CF, but in other diseases as well. And again, we're very interested in continuing the longitudinal sampling in a way to sort of age with our individuals that are on ETI therapy. And then potentially would be interesting to apply this to pediatric cohorts where no or mild clinically detectable disease exists, but transcriptomic signatures may be informative. And then with this, we're very interested in again, cycling back to this question of upper versus lower airway sampling. So just a few comments on this and the utility of this to lung disease. So basically this idea rests on what we know as the unified airway hypothesis, which states that sort of the lower, the nose and the sort of the lung kind of resident airways are one unit. This is sort of less of a kind of a phenomenon, but the idea is that there's common inflammatory processes along the way. And while the nose is far less well studied compared to the lung in CF, it has been established as a surrogate in kind of multiple studies. It's starting to have widespread use in other diseases, notably in asthma. And then when we've kind of like dug down into literature supporting common inflammatory pathways, it looks like maybe the message right now is that there's sort of distinct but overlapping processes in terms of inflammation in the microbiome. So we're very interested in starting a dual sampling project where we're going to be nasal brushing individuals prior to transplant or a medically indicated bronch. And then if anybody's interested in potentially exploring this in an animal model, I'd be very happy to talk about that. And then with that, I'm gonna say thank you again. Happy to take questions, and please check out my shameless plug for a postdoc position if you know anybody ready and able.
Good. Thank you very much. So we have time for a few questions. You can either type them into the app or it's a small room, just be clear. We can do it the old-fashioned way as well. Just.
Thank you.
Your transcriptomic data showed a lot of interesting stuff, but a lot of it was, I guess, maybe not unexpected with the inflammatory increasing. Was there anything in there that you found particularly surprising that's maybe a different ontology than you were expecting, or in your total transcriptomics, anything that was kind of out of the ordinary over the 6 months?
So, I mean, probably looking out to the two-year cohort, this potential for this transcriptomic resurgence was the part that was the most surprising, and this is why I kind of showed you all of our batch correction and all of our kind of due diligence data. What I would be spending most of my time currently agonizing over is how to interpret the upregulated signatures. And a lot of these speak to a very, very drastic metabolic remodeling. And this is where I think a lot of biology that may have been eluding me or maybe us as a community in terms of what is the difference between a chronically ill versus a slightly healthier airway epithelium, how that sort of rebuilds over time when you relieve the pressure of the extensive mucus. Yeah.
Okay. Any other questions?
Yeah. Yeah.
I think it's a similar question, but have you seen any pattern of normalization of the epithelium, like in terms of cell composition? Can you grasp that aspect of healing? Does the epithelium come back?
So this is why I kind of included those two highlights on the reduced cornification program and the increased ciliogenesis program. We also see a reduction in signatures that are maybe consistent with mucocell metaplasia. So very much we do see that. I'm going to talk a little bit more about that tomorrow, especially supported by the single cell, and then also showing the data from the cultures that are associated with this, which perform better, make a healthier epithelium in vitro at 6 months than at baseline. Yeah.
Great. Thank you. We should probably
Thanks
move
very
on.
much.
Thank you. Excellent talk. Our next speaker is Dr. Bradford Woodworth from the University of Alabama. He's going to talk about hypoxia-induced CFTR dysfunction and sinusitis.
There we go. Thank you, NACFC, for allowing us to present our research today. Some of my disclosures and research support. So with highly effective CFTR modulator therapy, this is noted to have a great impact on the sinuses in cystic fibrosis. I'm a rhinologist I'm an ENT physician who specializes in CF sinus research, but I also look at the way that modulators might impact regular sinusitis as well. When we went back to look at the goal data for ivacaftor, found that in G551D patients that this improved their the rhinologic, psychologic, and sleep-related quality of life on the SNOT-20 PROM. It's a patient-reported outcome measure. And then this was followed up with a study on highly effective modulator treatment, sinus symptoms, and it's basically showing that the modulators really impact the health of the sinus. Now, when you look at a regular CF sinus scan prior to CFTR modulator therapy, what you see is a sinus filled with mucopus. These are the maxillary sinuses underneath the eyes, the frontal sinuses here. And one can imagine, sorry. Thank you. One can imagine that this scenario where the maxillary sinuses are full of mucopus and the frontal sinuses, you have a condition where you have low oxygen conditions. Before you woke up today, I'm sure that you didn't get to think you were going to see the inside of someone's snot-filled nose. But this is a cystic fibrosis patient, the same patient, who is about 4 weeks on therapy, and that's the sphenoid sinus back there. This is the left side of the nose, and you can still see there's extensive mucopus and clustering of mucus within the sinuses, but the CFTR modulator therapy has actually cleared up a lot of the disease. And when we switch back, and he's been on the therapy for around 3 months, you'll see here, he's actually completely cleared disease without any additional antibiotic therapy, any topicals, anything new to his regimen. He's completely cleared the right side. The left side just has a little bit of residual mucopus and stasis on the sinus floor. And so these sinuses can remodel into normal epithelium, or at least functionally normal epithelium. The question is, though, there are some patients who, let's see, I'm looking here. There are some patients who actually, like this patient, responded really well, and this is a CT scan after 3 months of modulator therapy. This patient has been on CFTR modulator therapy for about a year and is not responding, even though she has an F508del mutation. So the question always is, like, what is creating the problem when these patients aren't responding? I mean, she's got some clearing in the ethmoid, but these maxillaries are still having problems. And one of the things that we think about is the low oxygen tension levels in the sinuses. And my friend Kasper Anius from Copenhagen in Denmark did a study back in 2011 looking at an oxygen probe in CF sinuses. And so he'd take these patients, they had CF sinusitis, he'd place it into an open, and remember, these are patients that have had operations, so they can't access the sinus without the operation. The probe down at the tissue level essentially showed at the tissue level near anoxia. So this shows that the cells at the surface level, when they've got tons of mucopus on top, they have issues getting oxygen to those cells. This is an area that we had a lot of interest in, and again, I study non-CF chronic sinusitis as well. But the dysfunctional sinus and the impact on mucociliary transport, there's overwhelming evidence that it's decreased, right? So saccharin transit times, we place a little bit in the nose or a little bit in the maxillary sinus, we see that there's a decreased saccharin transit times, as well as radionuclide tracers. There's also a decreased mucus strand velocity. So you take a patient like this who's got a dysfunctional maxillary sinus, and you take the excised mucosal explant, and they've got decreased movement out of the submucosal glands as a mucus strand. So this is really good evidence that there's a decreased mucociliary transport in chronic sinus disease. I think hypoxia has something to do with it. And we know that there's long-standing evidence that acquired CFTR dysfunction or deficiency is prevalent in a lot of models. COPD, we've heard it. There's been ivacaftor studies, as well as some other studies looking at CFTR activators in COPD. Hypoxia is a long-standing method, and then there's a bacterial toxin secreted by Pseudomonas aeruginosa, but also lipopolysaccharide, which is some of our work that we did looking at the way LPS actually impacts the ability to transport chloride. So this was back when I was a wee lad. It was like about 11 years ago in the lab, and so this is a short circuit current and oozing chamber of human sinonasal epithelial cells cultured on an ALI, and you can see they've got a decreased short circuit current with forskolin stimulation, both amiloride and forskolin, as well as you can see with INH-172, it is the CFTR-mediated current that's impacted. Everything's suppressed, but it really makes an impact when you look at what we call micro-optical coherence tomography, which is a technique developed by Steve Rowe and Mass General. And in this technique, we can look at simultaneous measurements of airway surface liquid, PCL, CBF, as well as MCT. And what we see is a decrease in all measures of mucociliary transport, ASL, PCL, CBF, as well as later data that I'll show with MCT. So it got me thinking when we did this study last year, it's published in Redox Biology, looking at LPS, and we discovered that LPS really decreases CFTR open probability and mucociliary transport really through a mechanism of probably direct damage with reactive oxygen species. So it stimulates reactive oxygen species, carbonylates CFTR, you end up with a decreased open channel probability. And our question was, well, the same mechanism. So we kind of revisited this. We also have a newer model of hypoxic rabbit maxillary sinusitis, which is really nice to study this in. So basically, the concept of the study is you have an in vitro arm and an in vivo arm. In vitro, we're using mammalian respiratory cells. We look at CFBEs and some others, but we're going to incubate those in hypoxic conditions, look at the Using chamber, but really want to look at patch clamps. So really with Using chambers, you're getting contributions from all sorts of other channels, and with a patch clamp, you get a really good view of what happens to the CFTR channel itself. And then we looked at fluorescence microscopy for reactive action species and micro-OCT, which is that technique for measuring mucociliary transport and the markers of mucociliary transport. And then with our rabbits, what we do is we block the osteometal complex. We'll show you how that we do that, and develop a rabbit sinusitis model. And that's for 2 weeks. And what we do is we confirm with micro-CT, oxygen levels, HIF1 alpha, which is an indirect marker, and pH change. And then really our outcome measures are sinus potential difference. So what you're looking at is actual measurement of chloride transport across the maxillary sinus epithelium, and then excised samples looking at micro-OCT to look at the mucociliary transport rates. So first off, looking at just regular murine nasal epithelia in hypoxic, using different levels of hypoxia, you can look at anoxia and you get a pretty large reductions in this, but we don't really feel like this is a physiologic means. I mean, there's really never a true anoxia at the level. And so 1% is one of those measures that we've always looked at. It turns out at 5%, you start getting a decrease in significance at 48 hours. And so we know that even at just levels of 5% oxygen, you can develop an acquired CFTR defect. This is our summary data over here. It happens with both the milleride and forcellin, as mentioned earlier. It appears that the milleride sensitive component is probably not as robust or as important as the forcellin sensitive component stimulating CFTR. So looking at CFBE expressing wild type CFTR, there are standard conditions. 24 hours is just easy because you start it and then look at it the next day at the same time. It's a 1% hypoxia, 1% oxygen. We see a nice reduction in CFTR-mediated current. Looking at whole cell patch clamps with CFBEs, 24 hours, we see a dramatic reduction in currents attributable to CFTR and forced stimulated current. So you see the IV relationship, the current voltage relationship here with the hypoxic cells basically having a very decreased current compared to the controls related to forced stimulated current. So this helped us establish, okay, well, with the LPS, there was a concept of looking at reactive action species, and we found with hypoxia, you actually get a release of reactive action species in a similar fashion. In the LPS model, it actually peaks at 4 hours, and it's a similar stress response here. And we see with lytic cells, this is a DCF dye, and this is the total cell count that we're looking at the different time points, 15 minutes, 1 hour, 4 hours, and 24 hours. We see this peak at 4 hours, and then it goes down. And well, if we look at single channel patch clamp data, so this is cell attached patch clamp, you have control hypoxia, and then control conditions with glutathione, and glutathione is a reactive action species scavenger. And what you see is if you incubate it with glutathione, you actually basically eliminate the impact of hypoxia. So you're not actually giving the cells oxygen, you're just taking away the reactive action species, and you've completely normalized the current. So it's pretty remarkable. So what about the cell physiologic data? So this is going to be your airway surface liquid, PCL, as well as the mucociliary transport rate. And these are images that we can derive and videos that we can derive from micro-OCT. And what you see is the control here, the yellow bar is airway surface liquid, the red is PCL. Same over here with hypoxia, you see a significant decrease and an increase back to relatively normal levels with hypoxia when it's incubated with GSH. And these are reflected in the summary data below. So finally, we looked at the in vivo model, and this makes a lot of sense for us because, think about it this way. When you get a sinusitis, what happens? You get obstructed ostia, right? So ostia is a closure of a sinus, or the opening of a sinus. And so say you get a viral infection, you get a swelling, the sinus closes, then you go on to develop sinusitis, then you have a problem, and you go to the doc in the box, get a steroid shot and whatever antibiotics and try and get rid of it. But the concept here is that with the hypoxic rabbit model, we're physically obstructing it with endoscopic guidance with a little sponge, and this is for around 2 weeks. And we confirm the rabbit sinus opacification, 5 rabbits in each cohort, with complete opacification of the unilateral side where the obstruction is in all rabbits. And then what we do is we use an oxygen probe, pre-sense oxygen probe. This is placed at the level of the tissue, and we see it's about a 5% oxygen level. And if you remember from our in vitro data, we see that 5% oxygen in vitro actually causes an acquired CFTR dysfunction just at 48 hours. And so this is 2 weeks of 5, and we have a 5% oxygen level at the surface, and we've got a good cluster here. And then looking at indirect data, relative values of hypoxic inducible factor, which basically goes up when your cells don't have good oxygen. So we see that there's a dramatic increase in the HIF-1 alpha as well. Rabbit sinus tissue pH also is decreased, which is consistent with acquired CFTR dysfunction. And then if we look at rabbit sinus potential difference measurements, so this is our rabbit, and we can drill a hole through the front and put in the tubing to the back wall. And we make the same area every time. And what you see here is this is the black line for an SPD looking at forced gland stimulated current and zero chloride. And this area right here is the stimulation of the amount of transepithelial chloride that's crossing, and akin to that. And in the red, this is going to be your sinusitis. And so in these rabbits, you've got a real suppression of the transepithelial potential. And so this basically shows that you've got an acquired CFTR dysfunction in those sinuses. And let's see, can I click on these? I don't know if I can click. Well, these are videos of a control and sinusitis. So these are explants of the lateral maxillary wall of the rabbits. And so we can take these, put it on the micro OCT, and look at the various measures. And with mucociliary transport, we've got a dramatic reduction in mucociliary transport and the ability to transport mucus across that area. So this just kind of confirms what we're looking at. So what does this mean for what's the upshot of this? So when you're looking at treatment, so we're interested in nanoparticle research. We do a lot of different types of novel drug delivery in our lab, and one of the things we developed is an oxygen-generating biomaterial for chronic rhinosinusitis therapy. And this is, again, it's based off the concept of hypoxia, right? So delivering oxygen to the sinuses. This is a calcium oxide and peroxide and a beeswax and a catalase. And so the catalase helps prevent the conversion to peroxide, and you'd end up with just water and O2, but this O2 release is going to help with any sort of hypoxia that's in the sinus. So that's the concept. When you use an oxygen-generating biomaterial, we had several different forms. When we just use CPO, there's a lot more peroxide. This is a way to kind of decrease the toxicity. But when we take those cells and we incubate them in hypoxia, so 1% hypoxia, you get a reduction, of course. But when we use, so this is the PBS, when we use the oxygen-generating biomaterial, we get a restoration or an increase in the forced gland stimulated current, just putting those biomaterials on the hypoxic cells. So kind of a neat little therapy and administration, and this would be envisioned topically in the sinuses. And finally, another area of research was with the support of the CFF on a research grant. This is our glutathione sodium bicarbonate nanoparticle matrix. So what this concept is glutathione is a pKa of around 3, so it's pretty acidic. And we always want bicarb or some sort of buffer in the mix, and so we overwhelm it with sodium bicarb nanoparticles, and this is a releasing curve for what's equivalent of 1 millimolar of GSH and 24 millimolar of sodium bicarb, and these can be released over time. And this is another concept of actually taking a nanoparticle and treating these cells that are hypoxic. And what we see is that we can restore hypoxic cells. Again, these are cells that have been incubated in 24 hours of hypoxia. And you can see the different measures here. in the sake of time, I won't go over everything, but basically we've got a massive stimulation of mucociliary transport when we actually have bicarb and GSH in the matrix. These are just showing that nanoparticles do mix in with live acute sinusitis rabbits when you put it into the sinus, and it has no impact on sinus or a lung histology. So in summary, I hope I've convinced you that hypoxia induces an acquired CFTR dysfunction by using chamber and patch clamp, and has a significant impact on sinusitis, both with a CF, when you have CFTR modulators, those patients that aren't responding, concept would be there might be some hypoxia-related obstruction, and further surgery may help those individuals versus using different types of strategies like nanoparticles secreting oxygen. And then MCT and other functional anatomy is affected by that. Glutathione does help reverse the effects, and these oxygen-generating biomaterials hopefully are a good potential therapeutic strategy when it comes to hypoxic sinusitis, both in CF and non-CF individuals. So I'd like to thank my lab and people involved in all our awesome research, and the UAB CF Research Center, and I'll take any questions. Thank you.
Thank you. We have time for a couple questions.
Yes, sir.
Wholesale studies, do they have physiologic ATP and the physiologic Second question is, do you have glutathione?
Yeah, so we
Can you repeat the question?
Yeah.
Just make it clear.
Yeah, there was physiologic ATP. We did not, the glutathione was there at all times during the hypoxic incubation, so it was constantly in the solution. Basically, the concept would be to, we're not looking at what happens when you give the glutathione after. You're looking at it, if you're incubating with it, then you're taking care of those oxidative issues that are causing the problem.
Scott?
Yes, sir.
I wanted to ask about really what is the oxygen tension level at the level of the epithelium. In the first probe, you said it went down to 0, but then later on, also because I think you would need that oxygen to generate the oxygen radicals.
Yeah. So the oxygen presense probe that Casper and Nias used, when you put it down onto the surface, those are the levels that we see, right? In reality, we probably don't have 0% oxygen there, right? That just doesn't make much sense. With our rabbits, for 2 weeks, we've got a 5% level at the level of epithelium. So I think that's probably a little bit more realistic. Again, those are also CF patients that have chronic inspissated mucus. It's a little bit different than an acute sinusitis rabbit model that's been obstructed for 2 weeks. So it's kind of a little bit of apples to oranges, but I think the more realistic scenario is that it's probably a little oxygen at the level. Yes, sir.
A technical question you point out. I noticed that you had TGFβ at the transcriptional level.
Yes.
As you may know, you also dysregulated post-translation, so the stability in high oxygen level as it's been degraded by the coaches. Have you measured the actual levels of
We have not. So this was more of a confirmation that the cells were hypoxic, and so we didn't explore it further beyond that.
Okay, great. Thank you very much. We'll probably move on.
Thank you.
Appreciate it. Okay. Our next speaker is Dr. Kenichi Okuda from the University of North Carolina, Chapel Hill. He's going to look at pathways regulating mucin and fluid secretion in small airways.
Yeah, thank you so much for the introduction and then this opportunity to present our data. I'd like to talk about a little bit pathways balancing basal mucin and CFTR-mediate fluid secretion in the human small airways. There is no disclosure to relate this presentation. So So while we recognize that multiple sites in the lung are affected in CF, such as superficial epithelia from large to small airways, and also submucosal glands, but except for alveoli. There has been solid evidence that mucus plugs occurs in the small airway region and associated with small airway dysfunction in CF. Yeah. So this, for example, this micro-CT imaging data shows the mucus plugs occurs at 6th generation bronchi to all the way down to the distal airways. And also, this in vivo clearance, particle clearance data shows the impaired peripheral particle clearance, while central lung clearance is comparable between normal versus CF. To understand why small airway regions are vulnerable and susceptible to CF lung disease, it is important to understand how small airway mucus homeostasis is maintained at baseline and how it fails in CF. So our previous morphometric studies identified some differences in mucociliary clearance-related parameters between large and small airways. This morphological analysis revealed a decreasing gradient of housekeeping mucin MUC5B gene expression from proximal to the distal airways with continuous and almost consistent expression of CFTR. Also, recent single-cell RNA sequence data, including our own, identified multiple cell types that express CFTR. That include ionocytes and secreted cell population and some of the basal population. And then our studies, especially focusing on small airway, identified secreted club cell as the most common frequent cell type that express CFTR in small airway region. So, but these data suggest there might be different mechanisms that regulate mucociliary clearance between large and small airway regions. Also, recent Nature paper showed small airway epithelia uniquely express some markers, including SFTPB and SGB3A2. So, and then these markers are exclusively express only bronchial epithelium, but not expressing these cartilaginous larger bronchus. And then to perform functional study and study small airway specific mucociliary mechanism, we have developed small airway epithelial cell in vitro cell culture model. So we isolated large airway and small airway epithelial cells from different each region, and then I expand them and then culture them in air-like condition using the exactly same media and the same condition. Then after differentiation, small airway epithelial cell cultures express these small airway unique markers, and which is consistent with in vivo normal human tissue sections. Then we have recently extended our ability to isolate and culture small airway cells from CF lung using the bulk digestion method, and the details are presented by Shannon at poster 423. And using this in vitro model, firstly, we have tried to characterize differential mucus biophysical properties and ion transport function between large and small airway cultures. So these are percent solids data. So the small airway epithelial cell cultures show lower percent solids compared to large airways. With the percent, the solid component is similar between large and small airway cell cultures. And the difference comes from this fluid secretion. Small airway epithelial cell cultures produce more fluid secretion. And then consistent with this data, Using chamber measurement shows like almost twofold higher CFTR response to force calling in small airway cell cultures compared to large airway cells, while epithelial resistance is similar between large and small. And also further consistent with these data, mucus viscosity is lower in small epithelial cell mucus, and which is associated with lower concentration of secretory mucins MUC5B and MUC5AC. But now the question is, what makes difference in CFTR function between large and small array cultures? Then we perform the single-cell RNA sequence to identify cell types that express CFTR in these large and small epithelial cultures. So we identified a unique subset of secretory cell population, this SAE secretory cell population here. And then this small array epithelial cell secretory cell population is characterized by small array cell markers, SGB3A2 and the SFTBB. But also, interestingly, relatively high levels of CFTR and also MUC5B. So this data suggests like this cell type may be important in the small array region because like this single cell type expresses both mucin and CFTR. And then they might regulate both mucin and fluid secretion as a single cell type. And then next, we have carefully looked at the differentially expressed genes in this small array secretory cell population and identified this iron receptor gene is highly expressed in this population, as well as some IL-1 pathway downstream genes here are including IL-6, CXCL1, 8, and then some chemokine CCL20, in addition to CFTR MUC5B. And also published evidence shows IL-1 pathways are one of the important pathways that regulate CFTR function and mucin production in human lung cells and also CAR3 cells. And then we hypothesized, and also we have confirmed this finding using single cell dissociation with RNA in situ hybridization. And these small airway secretory cells expressing SGB3A2 express co-localization of CFTR with IL-1 receptor genes. And based on these data, we hypothesized IL-1 pathway might regulate the baseline CFTR and also mucin secretion at baseline. Then we tested this hypothesis using the IL-1 receptor specific inhibitor in small epithelial cell cultures. Firstly, qPCR data shows significant reduction of MUC5B and also IL-1 pathway downstream genes CXCL8 and 1. So this data suggests even under baseline, IL-1 pathway is active in small epithelial cell cultures. And importantly, when we perform leucine chamber study, so this is this middle black line is a small epithelial cell culture control, and then when we treat the cells with IL-1 beta, phosphorin response is increased almost twofold, and then when we added IL-1 receptor antagonist, this increase is completely suppressed, and then interestingly, when we added IL-1 receptor antagonist alone, again, that CFTR function, phosphorin response is decreased to the half. So this data suggests under baseline, at least partially IL-1 pathway regulate CFTR function. But also we have noted, so there is still CFTR response remain after adding IL-1 receptor antagonist. That suggests there is another pathway that regulates baseline CFTR function. And then since this small array secretory cell express CFTR, so next we tested if this cell type can be a target for CFTR restoration. So we transduced wild-type CFTR using a lentivirus system under rat CCSP promoter to specifically transduce wild-type CFTR in CCSP-positive secretory cells in both large and small airway culture obtained from CF lungs. And firstly, we looked at the specificity of rat CCSP promoter. So this whole multi-immunochemistry shows nice co-localization of CFTR with CCSP in non-ciliated cells. This Western blotting data shows wild-type mature CFTR protein bands in large and small cells where CCSP promoter transduced CFTR, wild-type CFTR is transduced. And then when we do the Using chamber study, so both large and small airway cells, the CFTR function is restored, and then especially in small epithelial cell cultures, bigger restoration was observed. Then finally, because this small area secretory cell express mucin and CFTR, and then seem to be a key cell type that regulates small area mucus clearance, so we try to understand what regulates this cell type, and eventually distal airway mucus homeostasis. And we focused on NKX 2.1, which is a transcription factor required for lung development. And this transcription factor is expressed as a gradient from proximal to distal airways in adult airways. And also this is expressed in alveolar epithelial cells. And then we knocked out this transcription factor in primary large and small epithelial cultures. And then KO efficiency is pretty good, so more than 90% in all the codes we tested. And then we quantified major cell type, the frequency of major cell populations. And in large airways, there was no difference in frequency of major cell types. However, in smaller airways, there was a massive increase in the MUC5AC positive cells in NKX 2.1 KO cultures. And in contrast, SFTPB positive small airway secretory cells are significantly reduced in NKX 2.1 KO cells. So this data suggests it's possible the replacement of small airway secretory cells with MUC5AC positive goblet cell happens when we knock out NKX 2.1. And this HE staining shows goblet cell, morphologically goblet cell metaplasia in these KO cells. And then we speculate that this is really bad in small airways because small air mucus may be supposed to be low concentrated to be moved easily. But if this mucus goblet cell metaplasia happen and then more sticky MUC5AC increase, then small air MCC should be disturbed. And also another bad thing in NKX 2.1 knockout is the reduction of CFTR. So this might reflect the loss of this small airway secretory cell population in this NKX 2.1 KO cultures. So in conclusion, human small airway secretory cells express MUC5E and CFTR. Baseline MUC5E and CFTR-mediated fluid secretion is at least partially regulated by endogenous IL-1 pathway presumably in the small airway secretory cells. And the small airway secretory cells can be a cellular target for CFTR molecular therapy. And finally, so I like to show a big acknowledgement for all my collaborators, especially Dr. Boucher and Dr. O'Neill and Dr. Randall and also Dr. Button. So they have been giving me huge help for this project. And also I appreciate all the collaborators in UNC and the students and also lab members. And of course, I appreciate the funding to support this project from Cystic Fibrosis Foundation and the CFRI. Thank you so much for your attention.
Thank you very much. Any questions for Dr. Okuda? Paul? Yes.
Did you see a signal for oxide 1 positive ionized
In NKX 2.1 KO cells or just normal condition? Yes, I forgot to mention. Yeah, we have a few ionocytes, like rare cell population, including ionocytes, and then that population express typical ionocyte markers, and then they express the highest level of CFTR, and then that's consistent with all recent data.
So you had published that previously that there's a declining number of ionocytes.
Yes.
So your single cell in this culture model.
This time we Thank you so much. So yeah, we got a really few number, and then there was not so clear difference, but this time we didn't have clear difference, but very few numbers from n equals 3, much larger airway cultures. Yeah, thank you.
Any other questions? Use the app if you wish. All right.
Okay.
Thank you for your great
Yeah,
presentation.
thank you so much.
All right, our next speaker is Dr. Tahir Idris from the University of Geneva. He's going to talk to us about Wnt and TGF-beta signaling imbalance in CF airways.
Good afternoon, everyone. First, I'd like to thank the organizing committee for giving me the opportunity to present my PhD project. I'll be talking about the Wnt and TGF-beta signaling imbalance in cystic fibrosis area epithelium polarization. So I have no disclosure to, I have no relationships to disclose. So first, I will like to introduce the importance of the regeneration process of the area epithelium. It's a very crucial process that occurs after an injury to the epithelium, and it's important for maintaining the barrier function and as well as epithelial integrity. It's highly dependent on the proliferation and polarization processes, as well as the interplay between signaling pathways. So here I have an overview of the regeneration process after an injury to the epithelium. So first we have the basal cells that migrate and proliferate to cover up the injured area. This then creates an air-liquid interface that triggers the polarization and the terminal differentiation of the different cell types of the airways. And as I mentioned, there are several pathways that have been described in the literature to be important at the different stages of the regeneration process. And the interplay between them have also been described to be important. And if something goes wrong in this process, we can have problems with the structural integrity of the epithelium that's usually observed in cystic fibrosis. So our lab recently described the CF epithelium. We recently showed that the CF epithelium can be characterized by an overexpression, an apical redistribution of fibronectin and its receptor, the active form of beta-1 integrin, when normally they're supposed to be found at the basilateral parts of the epithelium. We also showed that this apical complex can serve as a docking station for bacterial adhesion. And since this situation is completely abnormal and resembles an epithelium that has a loss of cell polarity, we hypothesize that there's a defect in the polarization process of the CF area epithelium. So the aim of my PhD project was then to investigate the signaling event that regulates the polarization process of the CF area epithelium. As a first approach, we performed RNA sequencing on primary human area epithelial cells from CF and non-CF donors during the regeneration phase after an injury. Gene set enrichment analysis of the RNA-seq data predicted several pathways involved in proliferation and differentiation to be deregulated at the different phases of the regeneration process of the area epithelium. However, for the purpose of this talk, I will only focus on the Wnt and the TGF-beta signaling pathways. Gene set enrichment analysis also predicted several genes to be significantly enriched from these two pathways. And one of the most significantly enriched gene from the Wnt pathway was DKK1, which is a protein that's secreted and inhibits the Wnt signaling pathway. So we confirmed its expression by RNA-scope, which is a technique that allows you to fluorescently detect single mRNA transcripts of your target gene. And as we can see here in the CF epithelium, we have an increased expression of DKK1. Similarly, we also confirmed the expression of TGF-beta1, another gene that was also significantly enriched from the TGF-beta pathway by RNA-scope. And again, we have an increased expression of TGF-beta1 in the CF epithelium. So next, we wanted to see if we could confirm this on the cell model. And to do this, we used the CALO3 cells, and we knocked out CFTR with CRISPR-Cas9 to generate the CFTR knockout cells and its control. The cells were then cultured on filters and then passed to an air-liquid interface, and they were cultured at air-liquid interface for 14 days. And during this air-liquid interface, we have a dynamic balance between proliferation and polarization that's important in maintaining the structural integrity of the airway epithelium. So the first thing I did was to check if this balance was disrupted in the CFTR knockout cells. So first I monitored proliferation in our cells by doing a staining for Ki67, which is a marker for cell proliferation. So here I have confocal images at different days of air-liquid interface. And I'm showing the apical view of the epithelium where I had the most Ki67 staining. And here with the quantification, we can see that in the control cells, we have a peak in proliferation at day 3, and then a gradual decrease until day 14 of air-liquid interface. In the knockout cells, however, we don't have this peak at day 3. And what's also interesting is they stopped proliferating much earlier as compared to the control cells. So it seems by knocking out TFTR, it disrupts the proliferation process during air-liquid interface. Next, we monitored defects in polarization by doing a staining for apical fibronectin. So here I have confocal images. I have Z-stack images that's showing the top view of the epithelium at different days of air-liquid interface. And what we can appreciate is that in the control cells, no fibronectin can be detected. In the knockout cells, initially no fibronectin was detected. However, from day 3, we start to see some fibronectin that then continues until day 14 of air-liquid interface. Again, showing that we have a defect in the polarization process in the CFTR knockout cells. So next, we wanted to understand why this was happening. And to do this, we try to confirm the results that we found in the primary cells. So first, we performed our qPCR to monitor the expression of DKK1 in both cell types. And what we can see here is during air-liquid interface, we have an overexpression of DKK1 in the knockout cells. Likewise, we also monitored the expression of TGFβ1. And what we can see is at day 14, we have an increased expression of DKK1 in the knockout cells, confirming what we found in the primary cells. So next, we wanted to monitor the functional relevance of these results. So to do that, so because both proteins are secreted proteins, we incubated polarized cells with a physiological buffer for several time points, and then we collected what was secreted in this buffer, and then we measured DKK1 secretion as well as the effect on TGFβ signaling. So first, I will show results on the DKK1 secretion. So here we incubated with a buffer for several time points. And what we can see is after 1 hour of buffer incubation, we already have an oversecretion of DKK1 in the knockout cells that's completely absent in the control cells, suggesting that we have an inhibition of the Wnt signaling pathway. And to confirm this, we monitored the expression of beta-catenin, which is the main signal transducer of the Wnt pathway, at different days of air-liquid interface. So here with the quantification, what we can see is initially we actually have a similar level of expression between both cell types. However, from day 3, it starts to decrease until day 14, when it significantly decreases in the knockout cells. Again, indicating a decreased Wnt signaling pathway in the knockout cells. So moving on to the TGFβ pathway. So here we incubated with a buffer for 24 hours, and then we measured the effect on TGFβ signaling. So the first thing we did was we treated a reporter cell line that expresses SMAD binding elements to measure the SMAD response after treatment with buffer collected from control cells as well as the knockout cells, with and without activation with HCl. And what we can see here is in both cases we have increased SMAD response after treatments with the buffer collected from the knockout cells. What we also did was we treated control cells with these buffers, and then we measured TGF-beta signaling by monitoring phosphorylation of these mats. And again, we have increased phosphorylation of p-SMAD2 and SMAD3 after treatments with the buffer collected from the knockout cells, again indicating that we have increased TGF-beta signaling in our cells, in the knockout cells. So what I've shown so far is we have increased TGF-beta signaling and also a decreased Wnt signaling that creates this imbalance in the knockout cells. So what we wanted to do next was to see if we could correct this imbalance, and to do this we used pharmacological drugs. So the first drug we used was a Wnt pathway activator that blocks GSK3-beta and promotes beta-catenin accumulation to the nucleus and activation of the target genes. And the second drug we used was a TGF-beta pathway inhibitor that blocks the TGF-beta receptor, the TGF-beta receptors, and prevents the phosphorylation of the SMADs. So the first thing I did was I treated the cells with this drug, and as we can see here, we have an increase in beta-catenin expression in both the controls and also the knockout cells, as expected. Next, we wanted to see if there was a correlation between the activation of the Wnt pathway and the defect in cell polarity. And to do that, we monitored the expression of fibronectin. And interestingly, we have a decrease in the overexpression of fibronectin that's observed in the knockout cells here. And furthermore, we were also able to drastically reduce this apical fibronectin deposition that we observe in the knockout cells after treatment with the Wnt pathway activator. So it seems by activating with the Wnt pathway, I'm also able to abolish this apical fibronectin deposition, thereby restoring cell polarity. Next, we treated with the TGF-beta pathway inhibitor, and we also monitored the expression of fibronectin. And here we have a decrease in fibronectin expression in both the control and also the knockout cells. And with as with the previous drug, we also observed this decrease in the apical fibronectin deposition in the knockout cells after blocking the TGF-beta pathway. So it seems, again, by blocking the TGF-beta pathway, I'm able to restore cell polarity. So to conclude, what I've shown so far is we have this abnormal proliferation and polarization process in CFTR knockout cells that's due to an imbalanced Wnt and TGF-beta signaling. I also show that if I modulate both pathways, I'm able to prevent apical fibronectin deposition, thereby restoring cell polarity. And what's actually interesting is both the Wnt and TGF-beta pathways have been shown to positively regulate fibronectin expression in several contexts, including HEMT. However, in our case, we don't have a positive relationship between the Wnt and fibronectin expression. So we wanted to understand how the Wnt pathway was then able to regulate the apical fibronectin deposition in the knockout cells. And to do that, we plan to treat control cells with recombinant DKK1 to see if we can recapitulate the phenotype of the knockout cells on the control cells. And then we also plan to knock out DKK1 in the control cells to see if we can abolish this phenotype. And finally, we plan to investigate any possible crosstalks between the Wnt and TGF-beta signaling pathways to see if we can have more clues on how the Wnt pathway is able to regulate fibronectin expression in our cells. And with that, I'd like to thank everyone involved in the project. So the lab members, collaborators from the University of Geneva, the core facilities, as well as the funding organization. I'd like to thank you for your attention as well.
Thank you very much. Yes, Doctor. Yep.
So I'm wondering if you've looked at other outputs of cell polarity, either the cytoskeleton, like the cytoskeleton, or maybe basal complexes, or look at TDR as a barrier
Yes, yeah, actually we have checked all of them, and so we actually found disorganization of the actin cytoskeleton in the knockout cells. And we also found a lot of the junctional complexes that regulates the apical to basal polarity. They were also, we didn't check the power complexes. We checked the junctional complexes, like the tight junctions and the adherence junctions as well, and we saw that they were also defective in the knockout cells.
Yeah, you're fine.
Great job, thank you. I was asking if you would have a chance maybe to look at the localization of the TGFβ receptors, the ultra-polarization of these cells after CFTR knockout to see if that may also affect the signaling.
Yeah, that's actually it's an interesting, because we were thinking about it as well in the lab, like something that we were planning to do, and I think it's something that we will actually do, but we haven't done it yet, so it's something to do.
Prior to the start of the ALA differentiation culture, what media did you culture your cells in? Because some of the media out there contain Wnt agonists and TGFβ dual SMAD inhibitors, and I wonder if that has any effect if you tried different types of media before you switched to ALA. We didn't try different medium. We used the same medium for both. For both cell types. We used the same medium throughout the so before air liquid interface and also after air liquid interface. We used the same medium throughout, but we didn't. Because here we actually focusing on. We use the same medium for both cell types. So I don't think the composition of the medium will actually affect what we see here because we're looking at both cell.
In the back.
On the back.
During development, the Wnt TGFβ balance is correlated with the phosphorylation of the actin cells, so there's a little relationship. Paying attention to the role that play in the status of phosphorylation.
Yes, we actually have looked at EAP, and it's not something that I've looked at personally, but it's something that we've looked at in the lab. So I think it's another project that's actually that's ongoing in our lab, trying to characterize the localization of EAP in our cells as well, and we actually saw differences between both cell types regarding EAP.
Lane?
The cell line you use is the cancer cell line, right?
Yeah, they were, yes, they have cancer origin, yes.
The cell line you use is the cancer cell.
Yes.
So when you see knockout, actually it's knocking out, right?
It's actually a knockout because we use CRISPR to knock
And
out.
have you tried to do a rescue, for example, using the CFTR mutant to do a rescue, so the mutant will not be rescued of the type?
We haven't tried to rescue the mutant. No, we haven't tried yet. It's something that we plan to do. We try to look to use primary cells with different mutations to see if we can reproduce some of the results on all these different mutations. And we also plan to see if we can, if by rescuing these mutations we correct some of these defects as well.
Have you tried to confirm your results using the primary cell culture?
Yeah, we plan to do that. And also I've shown here that some of the results that we found in primary cells, we also confirmed on the cell line. But we also plan to do more studies on primary cells in the future.
Lane?
Thanks.
What's your hypothesis on abnormal increase? DKK1 CF?
Can you please repeat the question?
I was wondering what your hypothesis was for the abnormal increase in DKK1 CF.
That's it's an interesting question because it's something that we're also thinking about in the lab. We actually saw that we have more expression of DKK1 on the RNA level, and which can, which is probably the starting point into why we have this increased secretion in the knockout cells. So we think that we have more expression and possibly more secretion as a result of the increased expression of DKK1.
Thank you very much.
you.
Great talk. Final speaker is Dr. Joshua Donovan from University of Virginia, who's also going to talk about TGF-beta signaling and human bronchial epithelial cells.
So I'd like to begin by thanking the organizers for the opportunity to present our work on the novel mechanisms of TGF-beta signaling in the CF airway. I am Joshua Donovan from the University of Virginia, and I have nothing to disclose for this presentation. Now, we had a good intro to TGF-beta, but to do a deeper dive into its effects, it's a pretty potent extracellular cytokine involved in many intracellular processes, including cell growth, differentiation, organization, motility, and apoptosis. Specifically in the CF airway, it has four pretty negative effects that are important to keep in mind. The first is that TGF-beta is a pretty potent gene modifier associated with more severe lung disease in CF patients, especially in the most common CF mutation, F508del. In fact, 40% of F508del patients are high TGF-beta producers. Furthermore, there are a number of SNPs that are associated both with high TGF-beta levels and a more severe CF pulmonary phenotype. The next negative effect to keep in mind with TGF-beta is that it inhibits both the expression and function of CFTR, even in the presence of the newest triple combination of potentiators and correctors, elexacaftor, tezacaftor, and ivacaftor. Here we see Using chamber short circuit data demonstrating the rescue of CFTR in F508del cells by ETI, and then the subsequent inhibition by TGF-beta, even in the presence of ETI. TGF-beta also inhibits many other membrane channels, inducing mucociliary dysfunction in the CF airway. And the last important effect to keep in mind is that TGF-beta drives inflammation, goblet cell hyperplasia, and many other pulmonary abnormalities, both in the CF and non-CF lung. So these four effects put together, we can say that TGF-beta may represent an airway surface liquid inhibitor and an antagonist that limits the residual and corrected CFTR activity. So in light of these negative effects of TGF-beta in the CF airway, we wanted to do a deeper dive into the mechanisms of the TGF-beta activation pathway. So previous studies have shown that the PP1C, SMAD7, and GAD34 complex serves to inhibit the activation of the TGF-beta pathway by negative feedback. Now, the process by which this complex itself is inhibited, and thus allowing for the activation of the TGF-beta pathway, is unknown. But it is known to have many proteins that interact with it, including lemur tyrosine kinase 2, or LMTK2. In previous studies, we have shown that TGF-beta stimulation recruits LMTK2 to the plasma membrane. However, the process by which LMTK2 might impact this inhibition of PP1C and thus allow for the activation of the TGF-beta pathway was unknown. So our objective was to elucidate how stimulation with TGF-beta inactivates that SMAD7 PP1C negative feedback and allows for the activation of the TGF-beta signaling pathway in human bronchial epithelial cells. Now, all of these studies were performed in polarized human bronchial epithelial cells, either immortalized CFBE cells or F508del homozygous primary cells. So we began by looking at the initial activation of the TGF-beta pathway and in looking at the inhibitory phosphorylation of PP1C. So after TGF-beta treatment, we found a significant increase in the amount of phosphorylated PP1C, demonstrating the necessity of this inhibitory phosphorylation in the activation of the TGF-beta pathway. So in light of the importance of the PP1C complex in TGF-beta activation, we performed RNA sequencing after TGF-beta treatment to get a better understanding of the different regulatory elements of PP1C. And interestingly, we found that 26 genes that affect PP1C were affected, but most notably, SMAD7 and CDK5 were found to be upregulated. Now, SMAD7 upregulation is consistent with recruitment of PP1C, and thus the negative feedback inhibition of the TGF-beta activation pathway. Previous studies have demonstrated that CDK5 phosphorylates LMTK2, which then facilitates the inhibitory phosphorylation of PP1C, suggesting a novel role of LMTK2 in the activation of the TGF-beta signaling pathway. So, in looking at this potential role of LMTK2 on the activation of the TGF-beta signaling pathway, we performed LMTK2 knockdown and then RNA sequencing to try to get a better understanding of all the different points at which LMTK2 is involved in the signaling pathway. So after siRNA silencing, and we have the efficiency of our silencing there, we found that 33% of the genes in both the canonical and non-canonical TGF-beta signaling pathways were affected. Most notably, TGF-beta receptor 1 and 2 and SMAD7 were found to be downregulated. Interestingly, however, TGF-beta 2 and SMAD4 were found to be upregulated, suggesting that LMTK2 plays a role not just at the initial activation of the TGF-beta signaling pathway, but at multiple checkpoints throughout the entire pathway. However, looking specifically at the initial activation of the TGF-beta pathway, we wanted to see the effect of LMTK2 knockdown on the inhibitory phosphorylation of PP1C. So after siRNA silencing and TGF-beta treatment, we found a significant decrease in the amount of phosphorylated and thus inhibited PP1C. And this demonstrates that LMTK2 is required for the inhibitory phosphorylation of PP1C and thus the subsequent activation of the TGF-beta signaling pathway. Next, we wanted to look at a few proteins slightly more downstream in the canonical TGF-beta signaling pathway, SMAD2 and SMAD3. Now these proteins are phosphorylated by TGF-beta receptor 1 and 2, and then are translocated to the nucleus with SMAD4. So after LMTK2 knockdown and TGF-beta treatment, we found that there was no overall effect on SMAD2 activation, nor was there any effect on SMAD2 trafficking to the nucleus. However, under the same conditions, we found a significant decrease in SMAD3 activation and in SMAD3 trafficking to the nucleus, suggesting that LMTK2 specifically attenuates the SMAD3 pathway. So, in summary, the activation of the TGF-beta pathway is inhibited by negative feedback by the PP1C, SMAD7, and GAD34 complex. Stimulation with TGF-beta recruits LMTK2 to the plasma membrane. Well, once LMTK2 reaches the plasma membrane, It facilitates the inhibitory phosphorylation of PP1C, allowing for the activation of the TGF-beta signaling pathway, specifically through the SMAD3 mediated process. We've also shown that LMTK2 plays a role at many other checkpoints in the TGF-beta signaling pathway, and our hope is that through this better understanding of the mechanisms of the TGF-beta activation and signaling pathway, new therapeutic targets and pathways can be found that help to limit the negative effects of TGF-beta in the CF airway. And I'd like to acknowledge everyone who had a role in this, our principal investigator, Dr. Agnieszka Swiatek-Urban, our past and current lab members, especially Daniel Cruz, and our collaborator in Portugal, Dr. Carlos Farina. I'll just go back to this for you guys to take a look at.
Thank you. Thank you. Any questions for Dr. Donovan? Did you look at any functional outcomes, proliferation, PAA1 expression?
No, we did not. We focused mainly just on the different pathways, not on any functional analysis, other than some Using TER data. But
Okay. Did you look at any SMAD independent pathways? LMTK2 does quite a few things. TGF-beta can signal through multiple pathways, TAC1.
We focused mostly on the canonical TGF-beta signaling pathway, and specifically mostly on just SMAD2 and SMAD3.
Thank you very much. Any other questions? All right. Thank you for your presentation.
Thank you.